Alrighty, hello everyone. I am Shritika Yola, Chair of the NSGC Webinar Subcommittee. I want to thank you all for joining today's member webinar, Tricky Neurogenes, an Overview and Update. We encourage questions to be asked throughout the presentation. As a courtesy to our presenters, all attendees have been muted. Please utilize the Zoom questions box to type any questions you might have. Questions will be read aloud during the last 10 minutes of the presentation. Today's learning objectives are: indicate the primary disease mechanism for Rfc1 related canvas spectrum disorder and how genetic testing is performed. Evaluate the utility of optical mapping in the context of genetic testing for Fshd. Summarize the nuances of GBA gene testing as it relates to Parkinson's disease. And list the key features of SORD neuropathy. Today's speakers are Soma Das, Ph.D. Soma is a director of the Genetics Services Laboratory at the University of Chicago, where she has been the director since 1996. She is one of the pioneers of the molecular diagnostics of rare orphan genetic conditions. Our particular interest of hers is in the diagnostics of neurological conditions. Rojan Kavosh is a licensed certified genetic counselor and a genomic testing consultant at PerkinElmer Genomics. Prior to joining PerkinElmer Genomics, she worked as a perinatal genetic counselor in the fetal center at Stanford Children's Hospital. Michelle Rockman is a genetic counselor in adult neurology at Jefferson in Philadelphia, specializing in movement disorders. Michelle currently serves as co-chair for the Neurosig New to Neuro subcommittee. Tara Jones is a genetic counselor in adult neurology at Cedars-Sinai in Los Angeles, specializing in neuromuscular and dementia indications. Tara currently serves as co-chair for the Neurosig grant and webinar subcommittees. Her research interests include data-driven service delivery models to improve patient and provider outcomes regarding pre-symptomatic genetic testing. So that you have a little better idea of today's presentation, here is the layout. First, Soma will discuss Canvas and Rfc1. Then we'll turn it to Rojan, who will talk about FSHD1 and optical mapping. Next, Michelle will talk about Parkinson's disease and GBA1. And then finally, Tara will close out and talk about CMT2 and SORD. So to our first speaker, Soma, take it away.
Thank you. So my talk today is going to be on Rfc1 and canvas and spectrum disorders. And I am going to focus more on the methodology and some of the challenges with testing for this gene. Next slide, please. So our lab performs testing for Rfc1. Next slide. So CANVAS is an acronym. It stands for cerebellar ataxia, neuropathy, vestibular areflexia syndrome. The primary clinical features are cerebellar syndrome with cerebellar atrophy, sensory neuropathy, bilateral vestibular impairment, which results in some of the vertigo and the dizziness that's seen with this condition, dysautonomia, a chronic cough, and age of onset is often mostly in the 50s, later age of onset. It's an autosomal recessive condition, and in 2019, biallelic expansions within the Rfc1 gene were identified and seem to be present in approximately 90% of patients with clinical features or clinical diagnosis of canvas syndrome. It was also found to be present in, besides well-characterized patients with canvas, also seemed to be a frequent cause of ataxia in about 20% of sporadic late onset ataxia cases, and up to 60% in patients with additional features such as sensory neuropathy and bilateral vestibulopathy. And in 2021, last year, a study showed that about 30% of patients with idiopathic sensory neuropathy also had biallelic expansions of the Rfc1 gene. So repeat expansions in I'm sorry, I'll let you know, back to the next slide, back to the previous slide. So repeat expansions in Rfc1 have been observed in a spectrum of patients ranging from classic canvas to those with only some features, such as late onset ataxia and sensory neuropathy. Next slide, please. So Rfc1 codes for the replication factor C subunit 1, which plays a role in DNA replication and repair. Intron 2 of the Rfc1 gene has a pentanucleotide repeat of AAAG, which typically has about 9 to 11 repeats. And patients with Rfc1 disease have an expansion that ranges from about 400 to 2,000 repeats. But in addition to this expansion, there's also a change in the pentanucleotide unit itself. It changes from AAAG to AAGGG. A study of the normal population for this repeat region demonstrates that this region itself is polymorphic. The vast majority of the population have 9 to 11 repeats of this wild type AAAG. About 13% have a longer version of this repeat motif in about the 15 to 200 repeat range. A smaller percentage of the population have an alternate version of the repeat motif. It's an AAAGG, which is usually quite long in the 40 to 1,000 repeat range. And then there's a smattering of other repeat motifs that are not fully determined. And these all represent normal, non-pathogenic repeat motifs. And throughout my talk, I'll be depicting the non-pathogenic repeat motifs in blue and the pathogenic repeat motif in red to distinguish them better. A combination of studies, a number of different studies have estimated the carrier frequency of the pathogenic repeat motif to range from between 0.7 to 4%, with an estimated prevalence of this condition to be between 1 in 20,000 to 1 in 625, which for a Mendelian genetic condition is not that uncommon. Next slide, please. So in our lab, we perform testing for Rfc1 repeat expansion by repeat prime PCR. A repeat prime PCR allows for the detection of a specific repeat motif. And it's the same methodology that's performed for other repeat expansion disorders, such as the spinocerebellar ataxias, but with the added complexity that testing for more than one repeat motif is necessary for Rfc1. So there are 3 sets of PCRs that are set up for each sample. First, there's a flanking PCR, which is just a straightforward PCR, regular PCR across the repeat region. Products are obtained and their size for the repeats in the normal size range. If there is an expansion of the pathogenic allele, no product is obtained because it's too large to be amplified. The second PCR is a repeat prime PCR for the non-pathogenic repeat, and products are obtained that are specific for this non-pathogenic repeat motif, and they're sized. And the third PCR is a repeat prime PCR for the pathogenic repeat, and again, products are obtained that are specific for the pathogenic repeat motif. And in this case, the size of the products are expanded and they're too large, so we can't size them, but we do know that they're specific for the pathogenic repeat motif. I'm going to now demonstrate some of the complexities of Rfc1 testing. However, before doing that, I wanted to show you, and I was going to do that using some case examples, but before doing that, I wanted to show you what some typical results look like. Next slide, please. So here, I'm showing you 3 reactions per sample, okay? So as I mentioned already, there are 3 reactions per sample, flanking PCR, and 2 repeat prime PCR specific for the non-pathogenic and the pathogenic repeat motifs, respectively. And this slide shows typical results for normal individuals, with the example on the left of an individual with repeats in the smaller more common size range, and on the right, an individual with a repeat in the larger size range. So flanking PCR shows two alleles containing 7 and 11 repeats for the sample on this side, on the left. And for this sample, and for on the other side, the sample, they have repeats of 10 and about 109 repeats. And the repeat prime PCR for the non-pathogenic repeat motif shows that both alleles contain the non-pathogenic repeat motif. And the way repeat prime PCR works, a series of peaks are obtained where each peak corresponds to a repeat unit. Repeat prime PCR for the pathogenic repeat motif shows no amplification. Next slide. Here on the left-hand side is a sample that's heterozygous for the pathogenic repeat expansion. Flanking PCR shows a single allele that contains 10 repeats. Repeat prime PCR shows that this allele contains the non-pathogenic repeat motif. And repeat prime PCR for the pathogenic repeat motif shows the presence of an expanded allele, which is seen by the sawtooth pattern of peaks that goes on and is too large to size. Taken all together, one allele contains 10 repeats, and one allele contains an expanded pathogenic repeat that could not be sized by the flanking PCR. And on the right-hand side is a sample that's biallelic for the pathogenic repeat expansion, where flanking PCR shows no amplification, as does repeat prime PCR for the non-pathogenic repeat motif, and only repeat prime PCR for the pathogenic repeat motif shows amplification and expansion. Next slide, please. The presence of rarer alternate repeat motifs in RFC1, though, can make result interpretation sometimes more challenging. Next slide. So I'll start off with the first case example, which is a 70-year-old male with cerebellar ataxia, cerebellar atrophy, dysarthria, neuropathy, nystagmus, and no family history. RFC1 repeat expansion testing was ordered. Flanking PCR and repeat prime PCR for the non-pathogenic repeat motif showed no amplification, and amplification was obtained with the pathogenic repeat motif showing this expansion. These results are consistent with biallelic expansion of the pathogenic repeat motif and was reported in this way. Next slide. We then received a sample on this individual's wife, who was an asymptomatic 69-year-old female. And when we did the testing, we got the same results on her. Flanking PCR and repeat prime PCR for the non-pathogenic repeat motif showed no amplification. And we obtained amplification for the pathogenic repeat prime PCR, which again would be consistent with biallelic expansion for the pathogenic repeat motif, which was unexpected because this is an asymptomatic female. We don't expect this result. We then tested this individual for the alternate rarer non-pathogenic AAAGG repeat motif. Next slide, please. And this individual was found to have this repeat. So this individual has this alternate AAAGG repeat as well. So this individual is not biallelic for the pathogenic repeat expansion, but rather is a carrier for this pathogenic repeat expansion. So we therefore do reflex testing for all samples that appear biallelic for the pathogenic repeat expansion and test for this alternate non-pathogenic AAAGG repeat motif. Next slide. So the next case example is a 67-year-old female with sensory neuropathy and mild cerebellar atrophy. Flanking PCR demonstrated the presence of two alleles, one with 10 repeats and one with about 45 repeats. Repeat prime PCR demonstrated that the allele with 10 repeats contains the non-pathogenic AAAAG repeat. However, repeat prime PCR for the other two repeat motifs were negative. So how would we interpret this case? And the way we did was as follows. This individual has one normal allele with 10 AAAAG repeats, and one allele with 45 repeats for which the repeat motif could not be determined. So we interpreted this as saying, as this individual has at least one non-pathogenic repeat, the Rfc1 gene does not appear to be implicated in this individual's disease phenotype. I showed this case to illustrate that even though we may not always be able to determine the nature of the repeat motif of an Rfc1 allele, we still may be able to make some conclusions about the involvement of Rfc1 in a particular patient. Next slide. So the final case I wanted to illustrate is a 68-year-old female from Tonga. Tonga is a country in the South Pacific. This patient had ataxia at age 58, sensory neuropathy, Parkinson's, dysarthria, cerebellar atrophy, nystagmus, and a positive family history with 3 affected siblings. We did flanking PCR, repeat prime PCR for the 2 non-pathogenic repeat motifs, and repeat prime PCR for the pathogenic repeat motif, and we obtained no amplification for anything, nothing. This would be indicative of biallelic expansion or the presence of 2 large alleles of some other repeat motif that we are not able to test for. Next slide, please. So in 2020, there was this publication, or a couple of publications, of biallelic repeat expansions of a slightly different motif, ACAGG, seen in 2 Asia-Pacific canvas families and 1 Japanese patient. Our patient was from Tonga, which is a country in the South Pacific. So we designed primers, repeat prime specific primers for the ACAGG repeat and tested it out on this sample. Next slide. And found amplification for this particular repeat in this sample. So we conclude that these results are consistent with this individual being biallelic for the ACAGG repeat expansion. And we've had 2 samples in our lab to date that appear to be biallelic for this repeat motif. And both those patients were from the South Pacific or from the Far East part of the continent. Next slide, please. We have been offering Rfc1 repeat expansion testing clinically since October of 2019. And we offer this testing as a standalone test and as part of the ataxia repeat expansion panel test, where it is one of 13 genes, including other spinocerebellar ataxia genes. And I'm showing here the number of samples that we've tested to date and our positive yield. So we have tested 417 samples for the ataxia repeat expansion panel test, of which 28 samples have been found to be biallelic for Rfc1 repeat expansions, that's 7%, and 11 samples were heterozygous for Rfc1 repeat expansions, that's 2.6%. This yield of 7% for Rfc1 is actually higher than that of any other genes that are tested for this, that are present in this repeat expansion panel for the other ataxia repeat expansion genes. When tested as a standalone test, Rfc1 just by itself, we've tested 406 samples to date, of which 153 samples are biallelic for Rfc1, that's 38%. 39 samples were heterozygous, that's 10%. 52 samples were normal, and 4 samples were inconclusive, that's about 1%. And the samples that were inconclusive have mostly been cases where no amplification was obtained for any of the primers, indicating that there were expansions or large alleles present for some other repeat motif. We often get clinical checklists that are completed with samples that are submitted for testing. And in addition to late onset ataxia, sensory neuropathy, vestibular impairment, and chronic cough are very often indicated to be present in the positive cases. And many times a family history consistent with the recessive condition is also indicated. In our experience, the positive yield for Rfc1 of close to 40% is quite high when there is a clinical suspicion and when testing for just Rfc1 alone is ordered. Next slide, please. And finally, I just wanted to mention a very recent publication, or one that's just been accepted for publication, is the finding of truncating variants in some canvas patients who are heterozygous for the repeat expansion. In fact, we have identified 3 such patients in our lab who are compound heterozygous for a repeat expansion and a truncating variant. Sequencing of the Rfc1 gene is therefore now an additional test and indicated especially in canvas patients who are heterozygous for the repeat inspection. Thank you very much. And I'm now going to turn it over to our next speaker.
Hello, this is Rojan speaking. Thank you for having me here today to speak with you about advances in technology and diagnostic testing for FSHD, specifically by optical mapping. Next slide, please. My disclosures include that I work as a genomic testing consultant for PerkinElmer Genomics, where we perform testing for FSHD. And as such, I'll be speaking to the testing process as specific to our laboratory. Next slide, please. The main takeaway of my talk today is that FSHD can have potential implications for the entire family, and that's why it's important for patients to get tested and confirm the diagnosis. And optical mapping is the most efficient and accurate way to do so. Next slide, please. To start with some background, FSHD is the third most common form of muscular dystrophy, and there are two types. FSHD type 1, which accounts for about 95% of cases, and FSHD type 2, which accounts for about 5% of cases. Next slide, please. Both have the same clinical presentation, and the only thing that distinguishes them is the genetic change that is causing symptoms, which is what determines a diagnosis of FSHD type 1 versus type 2. We're starting to learn more about the varying presentations based on the nuances of the genetic changes within each type. But for the purposes of my talk today, we're going to focus on the genetic distinction between these two types to discuss how we go about testing for them. DUX4 is a gene that is turned on or activated in FSHD. It's located in the D4Z4 repeat region of chromosome 4. We've learned that we need DUX4 to be turned on during very early stages of life, but when it remains turned on all the time, the protein product of DUX4 is toxic to muscle cells. As we know, our cells have different ways of turning off genes that don't need to be activated all the time and in all the cell types of our body. And one way cells do this is by methylation, where a specific chemical marker is attached to the gene and it tells the cell to keep the gene off so the protein product can't be made. If methylation does not occur, aka hypomethylation, the gene isn't being turned off as it should. In FSHD type 1, DUX4 stays turned on because there's a fewer number of D4Z4 repeats, specifically less than 11 repeats. So methylation cannot occur to turn off the gene because of this D4Z4 contraction. One click, please. In FSHD type 2, there's a normal number of D4Z4 repeats, but there's a change in the Fshd gene that inhibits its proper protein production. One click, please, which in turn interferes with methylation. So again, methylation cannot occur. For either type of FSHD to occur, the haplotype 4qa must be present on the chromosome. Next slide, please. Given that FSHD has overlapping symptoms with other genetic conditions that have different implications for medical management, outcomes, or inheritance, it's important to get an accurate diagnosis, and this has been challenging in the past. Next slide. Traditionally, testing for FSHD has included complicated algorithms, usually starting out first with Southern blot to estimate the D4Z4 repeat sizes, followed by haplotyping, then methylation studies, and finally Sanger sequencing or next generation sequencing of the Fshd gene if needed. Next slide. Of course, from a clinical standpoint, multi-step testing approaches like this can be complicated, costly, and time-consuming. So PerkinElmer has adopted a new approach to FSHD testing that makes the process less complicated and more affordable as well. We use whole genome optical mapping, where we isolate large fragments of DNA, upwards of 150,000 base pairs in size, from a whole blood sample. For reference, typical next-generation sequencing uses very small DNA fragments of about 100 to 200 base pairs. So these are much larger pieces of DNA than that. Saliva swabs and even older blood samples are not ideal for getting the size of DNA fragments, since DNA denatures over time when it's outside of the body, as we know. So those would not be good sample types for FSHD testing. Fluorescent probes are applied to these large DNA fragments, arranged every 6 base pairs or so, So that's approximately 500 million probes per genome. Another reference point that might be helpful, a microarray uses a few 100,000 to a couple million probes. So this is many more than that. The large DNA fragments labeled with their probes are then passed through very tiny tubes in the analyzer, tiny enough where only a single strand of DNA can fit into each tube to be read at a time. And in this way, we're able to linearize and take high resolution images of each strand. Lastly, we use a process called de novo gene mapping without using a reference genome to visualize the haplotype, whether it's A or B, the repeat number of the D4Z4 region within one repeat size, and any complex or balanced rearrangements. So the reason we're able to use optical mapping in this way is that the probe pattern or fluorescent pattern is unique for each chromosome, similarly to how a karyotype banding pattern is unique to a chromosome, for example. And that's what we are mapping here. In other words, we're taking very high molecular weight pieces of DNA in real time and mapping their probe signal using a fluorescent microscope to identify the chromosome and the specific banding pattern for the haplotype. Next slide. So the results that would be reported from FSHD type 1 testing, which is where we use optical mapping, would include the haplotype and the D4Z4 repeat size. And we've discussed how both of these are relevant in diagnosing FSHD type 1. Next slide. For FSHD type 2 testing, we use next generation sequencing to read through the SMCHD1 gene and look for harmful changes in the code that could disrupt gene function and contribute to the cause for FSHD type 2. Again, if someone also has the A haplotype on chromosome 4q. Next slide. If we test for FSHD types 1 and 2 together, the results would provide a full picture of whether either one is present. An additional note to mention here, since FSHD can have overlapping symptoms with other neuromuscular conditions, sometimes a diagnosis can be difficult to distinguish, and a provider may want to include testing for a larger panel of potentially associated genes in addition to testing for FSHD types 1 and 2. Our test menu includes an option for this via a broader panel, and in the same way that we use next generation sequencing to read through the SMCHD1 gene, we're able to efficiently look at multiple genes at once without any changes to turnaround time. So this may be something to consider when researching testing options at any lab you're considering ordering from. Next slide. So who should consider genetic testing for FSHD? If a patient or their child is suspected to have a muscular dystrophy and hasn't had genetic testing, if their current clinical diagnosis does not seem to fully explain the symptoms, or previous genetic testing did not provide a diagnosis, or if they have a family member diagnosed with FSHD, testing can be considered. Thank you, and I'll pass it on to the next speaker.
Michelle, you muted.
Great, that would help to unmute. Hi guys, I'm Michelle Rockman. I am a clinical genetic counselor specializing in movement disorders at Jefferson in Philadelphia. I do not have any disclosures to announce. As I started seeing patients with Parkinson's, I began to understand the nuances of the GBA gene. As I'm sure many of you are aware, biallelic pathogenic variants in GBA causes Gaucher disease. Also, heterozygous pathogenic variants in GBA cause a person to have an increased likelihood to develop Parkinson's disease at some point in their lifetime. Essentially, Parkinson's disease occurs when brain cells that make dopamine stop working or die. And dopamine is that neurotransmitter that coordinates movement and feelings of motivation and reward. So essentially, when dopamine cells die, we start to see those Parkinson's symptoms emerging. The most hallmark symptoms of Parkinson's, though they can vary, include tremor, bradykinesia, muscle rigidity, speech and cognition changes, as well as a host of other things. Next slide. GBA is actually the most common genetic factor associated with Parkinson's at around 3 to 7% of all adults with Parkinson's. And that number is a lot higher amongst the Ashkenazi Jewish ancestry at about 20%. The GBA gene, as it relates to Parkinson's, is autosomal dominant and has incomplete penetrance. The carrier frequency is 1 in 14 for Ashkenazi Jewish ancestry and ranges up to about 1 in 220 for those other populations. And what I want to talk about today is the fact that GBA has a pseudogene called GBAP1. Next slide. So pseudogenes are non-functional segments of DNA that resemble functional genes. And this is not something that's specific to Parkinson's or even neurology. We can see pseudogenes in the entire genome. For example, there's a pseudogene for a common cancer gene, P10, called P10P1. And normally, processed pseudogenes only have exons and they don't have introns. Therefore, labs are able to use primers in those introns to differentiate the pseudogene versus the functional gene. The problem occurs when GBAP1 actually has exons and introns. And also want to note that recombinant pathogenic variants between GBA and its pseudogene, GBAP1, really complicate this analysis further. Next slide. Therefore, the technique that is used to analyze the GBA gene is really critical. So from what I've seen, most laboratories that offer testing for GBA, they use next-gen sequencing for their Parkinson's panels. And the problem with that is that GBAP1 is a very common homologous pseudogene. So if the reads are a better fit for the pseudogene, they align to the pseudogene and not the functional GBA gene. Additionally, I've noticed that some laboratories will only offer targeted variant analysis to look at those most common pathogenic variants in GBA. And lastly, I have seen that some laboratories choose to only sequence certain exons. It's known that exons 9 to 15 are often highly recombinant and difficult to analyze. So certain laboratories will exclude that from their analysis because it's technically difficult to do. Go back. Thank you. So, however, there are some labs that take a different approach to GBA testing. And these laboratories, I've noticed, they use Sanger sequencing with long-range PCR and to distinguish the GBA gene from the pseudogene. So they rely on a few nucleotides that differ between the gene and the pseudogene. They also use reverse primers that are non-specific to allow the detection of recombinant alleles. And as a note, only a few labs use these techniques, and I have found the most success with GeneDx. Next slide. I added this screenshot in here to just illustrate this point that not all Parkinson's disease panels are created equally. This is a screenshot from a website, a free website that I like to use. It's called Concert Genetics. It kind of compares different laboratories' tests in a nice visual picture. So on the left, we can see Lab A. Their technique is deletion, duplication, and sequencing. Same with Lab B, but if you look at the mechanism of Lab B, they're pointing out that they use Sanger sequencing. And these two images here, they're showing very similar Parkinson's disease panels. You can also see that Lab B offers a bunch more genes than Lab A. So it's a nice visual picture to see in green which genes are common amongst the two panels and which genes are unique. Next slide. So in conclusion, my take-home messages here are threefold. So firstly, variant analysis of GBA gene is complicated by the presence of that highly homologous pseudogene, GBAP1. Secondly, It's important to communicate with the lab before ordering a Parkinson's panel to see if GBA has really good coverage. So you should be asking the lab what technologies are being used. And you can also check the test information sheet online. I know some laboratories have one of those. And thirdly, sharing is caring, and we all know that things are changing, especially in neurogenetics. So please update the Neurosig if you find out any new information on this topic. We would be grateful. Thank you. Now I will pass it over to Tara Jones to present her talk on CMT2 and SWORD.
Thank you, everyone. I'm very excited to discuss and refresh some of you on a novel gene that was just published back in 2020, which is SWORD, and it has actually been identified to be the most common cause of recessive CMT. Next slide. I am a shareholder of Invitae and Myriad. Next slide. Okay, so just an overview for Charcot-Marie tooth disease. It is an umbrella term for hereditary neuropathy. Most commonly, it is both sensory and motor, but there are subtypes that have pure sensory, pure motor, or sensory neuropathy with autonomic neuropathy. Generally speaking, CMT presents with symmetric length-dependent changes beginning in the toes, crawling up to the knees, and may also start affecting the hands, starting at the fingers. It is prevalent in 1 in 2,500 to 1 in 1,200 individuals. Back one slide. And the subtype refers to the inheritance pattern. So you'll see CMT1A, CMT2B, CMT4C. The inheritance pattern CMT1 and 2 typically refers to as autosomal dominant. CMT4 is typically autosomal recessive, and CMTX is typically X-linked. It also refers to the pathophysiology, whether the subtype is demyelinating, axonal, or intermediate between the two. CMT type 1 is typically demyelinating. CMT type 2 refers to axonal, and then CMT type 4 and CMTX can vary. And then we have the causative gene, which is the final letter of the subtype. So CMT1A is autosomal dominant demyelinating CMT caused by duplications in the PMP22 gene. A being meaning that it was the first gene to be discovered linked to the subtype. Next slide. So CMT type 2 often is difficult to identify the causative gene. So we're talking specifically axonal CMT. Diagnostic yield is less than 40%. This diagram here is representative of all patients that had CMT that was not type 1A. So they ruled out the most common subtype of CMT. And this was at 2 centers, University College London, who performed a 50 gene panel test, and then University of Iowa that kind of had varied panel tests between 18 and 135 genes. And we can see here that there was just about 22% of people with axonal/intermediate CMT that had an identified genetic cause. Next slide, please. And so in 2020, SWORD was discovered. This is a common cause of autosomal recessive CMT, and the gene codes for sorbitol dehydrogenase, which in normal cases is supposed to break down sorbitol into fructose. This C.757 del G frame shift mutation is the most common variant, and about 3 in 1,000 people are carriers for this. It accounts for 10% of undiagnosed CMT2 or distal hereditary motor neuropathy cases. And the average age of onset is in the late teens. Individuals can have purely axonal CMT with sensory and motor symptoms. They can have intermediate CMT with sensory and motor symptoms, or they can have a pure motor phenotype. And a majority of individuals have mild severity. All patients with SORD neuropathy so far have had motor involvement, and about half of them have had sensory involvement. In addition, all patients are found with increased serum fasting sorbitol. However, this is not a lab test available clinically, so it does have to be done under research basis. Next slide, please. Kind of piggybacking off of Michelle's talk, SORD does have a pseudogene, and it is in close proximity to the gene itself. It is thought to have arisen from a 0.5 megabase duplication of SORD. And is interestingly, 95% of control chromosomes carry that same common pathogenic variant in the SORD gene. However, this particular variant, if present in the pseudogene, is what's blocking translation and keeping the gene from actually being functional. Next slide. And so now that we know if we end up seeing an individual with CMT onset in their late teens, maybe early adulthood, with no family history, the likelihood of it being SORD is quite high, especially if they've had negative genetic testing to date. Unfortunately, it is not available on any clinical panels to this date. However, GeneDx is able to report on this gene on exome analysis when it is ordered for the indication of hereditary neuropathy. And then there are many other commercial labs that are working on validation for this. It does appear that the most efficient technology to be able to distinguish between pseudogene and real gene is Oxford Nanopore Tech long read sequencing. And there is a pharmaceutical company, Applied Therapeutics, that is offering serum fasted sorbitol testing to screen for candidates. Individuals are eligible if they are 16 or older with CMT or distal hereditary motor neuropathy of unknown subtype. There is a caveat that if a patient screens positive, the only way that they would be able to get their own genetic testing is if they are eligible for the clinical trial that AT is conducting, and are willing to move forward with being a subject in that trial. And so AT is doing a clinical trial on an aldose reductase inhibitor, which is upstream of the deficient sorbitol dehydrogenase gene, and it prevents breakdown of glucose into sorbitol. And that completes my portion. I will pass it over to Shrutika, who will begin the question and answer.
All righty. Thank you so much to all of our speakers for some very good information on all of these tricky neurogenes. We've had a lot of good questions come in for all of you. I'm going to stop sharing my screen a little bit so that we can see everybody, and then I'll direct some questions to each of the speakers. All righty. So to start, Soma, some questions that came in for you. First and foremost, what is the carrier frequency of the Rfc1 polymorphic expansion in the general population? Presumably, all of the samples presented were symptomatic for Canvas.
Yeah. So the first, when this was first studied, the first publication that came out on this did look at that, and the it was, I think it was about like 0.7% when they're looking at the frequency of the pathogenic expansion of Rfc1 in the general population. It was about I think that's right, about 0.7% of the general population had this pathogenic repeat expansion of Rfc1.
Okay. And then another question that came in, could the large yield for RFC alone be due to those patients already having other ataxia expansion disorders ruled out?
That could definitely be part of it. That could definitely be part of it, because Rfc1 has only recently been identified, right? This gene was identified in 2019. So prior to that, obviously, this was not been tested for. And there could be these patients who've been out there, who have not been tested, who have been tested for other genes. And only now are we doing the testing for this gene. So clearly, there have been patients who have been waiting for this testing to be done and are now being identified with the test being available. So part of this high yield could be due to the fact that there are these undiagnosed patients who have been out there, who are only now being identified because of the test being available. So that could contribute to part of the high yield that we are seeing. We've been seeing this. I mean, we've been doing this testing since 2019, but this yield has held. So even now until 2022, when I did this, even initially when we were doing this testing, this was the yield we were getting, and even now we still get the same sort of yield. So it hasn't dropped. So it still seems to be pretty consistent.
All righty. Thank you so much, Soma. Some questions for Rojan. With the new testing strategy for FSHD, does this change the need for multiple generations to be tested in order to perform Pgtm?
That's a really good question. As far as I'm aware, there aren't labs that are doing Pgtm by optical mapping. I think it depends on what the method is that they're using for testing. In terms of getting a diagnostic result, multiple generations don't need to be tested, but if the Pgtm is being done using linkage analyses, then that changes the answer. So I'm sorry, I don't have a good answer for that because our lab only is validated to test on whole blood. So I can't speak to the Pgt side of things.
No.
I see a little thing that says Tara would like to answer the question. Did you have something to comment on that too?
Oh, no, I don't actually, but I would agree with you, Rojan. I don't know of any labs that are doing optical mapping for that, but I feel like in most cases, any Mendelian disorder, we're going to be looking at needing not only the parent sample, but also their parents to do linkage analysis.
Let's see. Another question came in for you, Rojan, from Maureen. I believe the FSHD1 reports from PerkinElmer mentioned the translocations from chromosomes 4 and 10 are not detectable. Could you comment on this and what this means for clinical interpretation of results? Could this result in a false negative result?
Yes, so this is a really good question, and the answer is a little bit detailed. The short answer is yes, and I'm happy to discuss it further offline, but essentially D4Z4 repeats are located on chromosomes 4 and 10. Only the permissive haplotype on chromosome 4 contains a polyadenylation signal that stabilizes the DUX4 mRNA transcript, and there is approximately 98%, maybe 99% homology between the 10qa haplotype and the 4qa haplotype. And so there is, in rare cases, it is possible that the patient carries an actual pathogenic translocation, in which case follow-up investigations would be recommended based on the clinical picture and what other testing has been done. Pulse field gel electrophoresis would be the test that would be recommended to investigate the possibility of translocations, but optical mapping wouldn't be able to detect that because of the limitations I described. And again, I'm happy to go into more detail. That was kind of a truncated, short answer.
All right, couple questions for Michelle. Somebody is curious about how you think full GBA sequencing versus capturing the most common targeted variants, given the large proportion of people who will have one of these common variants. Do you always send for full sequencing?
Yeah, sure. I think that's a really fair question. I think you're getting at a great point that yes, if we're doing targeted analysis, we're going to capture most, right? So that's good, but is it the best? No, I don't think so. I think if you want to be as comprehensive and not falsely reassure your patients, then yes, I do always send for the best test that has the fullest coverage of GBA. I think it's important to remember that we're talking about neurology genetics, which is fairly new. And so we might not be aware of all the variants that are important to be looked at yet. So I just like to be sure and send for the most comprehensive test. Though you are correct, capturing the most common variants is good. It's a good plan. But no, I send for as much coverage of the GBA gene as I can.
Okay, here's a question for Tara or for anyone else who wants to answer. What literature databases do you use to help find published cases around these genes?
Good question. I'm part of the Inherited Neuropathy Consortium, and so it was our group that identified the SORD gene. So I was already privy to the information before it was officially published. I don't know. I think Soma might be able to answer that, particularly about Rfc1, because their lab is probably constantly paying attention to any new publications on Rfc1, but I do not actively search for anything on SORD.
Yeah, I mean, so yeah, right. So for Rfc1, yes, I mean, we do just search the literature on PubMed, just go onto PubMed and just do searches for Rfc1. I am in touch with the research with one of the research groups anyway that does work on Rfc1. So I have so I am in contact with them that way. But otherwise, I think just basically doing literature searches on PubMed using the gene name in question and just doing searches that way is the way I keep on top to try and see what new literature is out there for the gene in question. And that's pretty good, just doing PubMed searches for the gene in question.
All right, another question for everybody, unless anyone else wants to add on to that last question. Then moving on, for genes complicated by pseudogenes, if the lab starts with next gen and finds a variant, can they follow it up with another method then to see if it is in the real gene versus pseudo as needed? And I think this is a question we have across all clinical fields, not necessarily limited to neuro.
Yeah, I can take a stab at that one. So yes, typically long-range PCR or other validation methods can be run, but it will depend on the lab, whether they can do that in-house or if they would refer somewhere else that would do the additional analyses.
All righty. One other question that came in for Michelle. If patients are identified as GBA carriers as part carrier screening, so not diagnostic workup, are they being routinely counseled about the increased risk for Parkinson's disease associated with being heterozygous?
Let me make sure I understand the question. So if people who are getting carrier screening, who are thinking about becoming pregnant, they screen positive for GBA because GBA is on there for Gaucher.
Exactly.
If they screen positive, are they then being referred to someone like me in neurogenetics to get counseled for their risk for Parkinson's? That's the
I say yes,
Okay.
as well as the possibility of even having that discussion within the prenatal genetic counseling session.
And that's a wonderful, wonderful question. So at our clinic, we've decided to partner with the genetic counselor at our institution that works in prenatal. So me and her work really closely, and she is well aware of the risks of GBA and Parkinson's. So she has started referring some of her patients over to me to just talk about it. And see if, and leave it kind of up in, she'll counsel to say like, yes, you have an increased risk for Parkinson's. Would you like to open this discussion further and be referred to neurogenetics? So I think this kind of link between specialties is unique, but yeah, that's what we started doing here at Jefferson, because we do think that's important to open that discussion up if the patient is ready to talk about it, right? Because we're talking about someone who might be really young getting pregnant, and an adult onset condition. So you have to be really, really careful in your counseling.
All right, thank you. Let's see.
And
oh, go ahead.
I can also add to that. I actually did my thesis in grad school on this topic of whether it's being discussed by prenatal genetic counselors. And this was done back in 2017, but at the time, a lot of our participants kind of learned of the link through my survey. But there was a lot of discussion on the fact that there's so many conditions on carrier screening panels that it's impossible to really discuss during pre-test counseling, aside from maybe a brief statement about how learning of carrier risk may teach you about, some risks to your own health. But I don't know, I've never at least received any referrals for patients that end up testing positive for GBA. It's usually more of a concern in families that actually have Parkinson's, where they're familiar with the condition. Otherwise, because it's incompletely penetrant. I don't know what your experiences are, Michelle, if you've noticed that there are a lot of people who are really fearful of those results, but I feel like we can usually give them a lot of comfort in knowing that the risk is not anywhere close to 100% lifetime risk to develop Parkinson's.
Yeah, I would agree with all of that.
All righty. Well, I'm going to share my screen again, just as we wrap up in the last few minutes here. Before I close out today's member webinar, I wanted to turn it back over to Michelle for some announcements from the Neurosig.
Yeah, thank you. So there is a subcommittee amongst the Neurosig called New to Neuro, open to anyone who wants to join. And we would ask that anyone on this call, anyone who's a genetic counselor, who's interested in student contact to take this survey, you can scan the barcode with your phone, or you can go to this, this link. I tried to make it a shorter link for you guys. And the survey is just going to ask you a couple questions regarding if you would be open to even like answering questions from students, if you are open to hosting a rotation at your institution or wherever you work, or even if you would like to participate with a student on a thesis project. So any contact that you're willing to do, please fill out this survey. The New to Neuro group is just trying to collect this as a resource to use in the future. Thank you.
Perfect, thank you. And then one last announcement from the Education Committee. Our counterparts with podcast are planning to do an episode focused on the All of Us research program and their genomic results return plan and process. So keep your eyes and ears open for that. And with that, that concludes our member webinar today. Thank you so much to our speakers and the Neurosig for a wonderful and very informative presentation. This recording will be posted within 48 hours to the webinar page on NSGC. Have a great day, all.